All right, so I will go ahead and get started. I know people are going to roll in a little bit more, but hello everyone. Thanks so much for joining us here at NeuroHackademy 2020. And thanks again to Ariel and Tal for organizing this event and moving everything to the virtual domain this year. Since we don't have the opportunity to meet in person, I just wanted to start off by telling you a little bit about myself so that I can be more than just a non-specific Zoom character to you. As I just mentioned, I will try really hard to monitor questions as I present, but I am sort of bad at that. And I'm also not sure where that shows up on my display. So in case I miss your question or give a confusing answer, my contact information is provided here. So let's chat about me. I'm Angie. I am a mom to 3 boys. Including 11-year-old identical twins and a 2-year-old. I talk about my kids a lot because I'm like 10% a narcissist and 90% to normalize parenthood and academia. My family is still locked down and quarantined here in South Florida, and it is a righteous daily struggle to keep everybody busy and entertained. The work-life balance for us and for many families is a flat-out farce these days in the age of Corona. I am a professor of physics at Florida International University in Miami, and I am the founding director of our Center for Imaging Science, which hosts a 3T Siemens Prisma. Most of my research focuses on neuroimaging meta-analysis and the development of neuroinformatics and meta-analysis tools and strategies. I also spent a considerable amount of my time contributing to the ABCD study as one of their site PIs. So let's see what else. I want to tell you how much I love folklore, but I haven't listened to it yet because I've been too busy consuming Gaslighter on steady repeat. I'm feeling a little bit nostalgic. Tomorrow is my 45th birthday and has me doing lots of reflecting about my life and my career and my family, where I've been, where I still want to go. And so that's my segue to what we're going to talk about here today, which is meta-analysis and reproducibility. Reflection is a theme in my research, as meta-analysis is sort of a quantitative reflection process. If you'll recall, during Russ's lecture yesterday, he introduced reproducibility from a meta-analytic perspective by telling you about the Mueller et al. Null results that examine spatial convergence across depression studies. And I'd like to start back even further. Before we had this term reproducibility crisis, there were a good number of researchers who were simply interested in establishing consensus in measuring this to identify regions of consistent activation across studies. The first function location meta-analysis that I'm aware of was published in 1993 by Tomas Paus and colleagues. In that paper, they looked at various functional PET experiments that had been done that required hand responses, speech responses, and eye movements. And then they mapped the variability of those response types across the cingulate cortex. And this was followed by a similar effort in 1996 by Picard and Strick. And, you know, I find it really interesting that we've all been interested in medial frontal cortex for quite a while now, dating back to farther than a lot of people realize. So meta-analysis is really kind of different in neuroimaging. And instead of looking at significance effects meta-analysis, we really tend to focus primarily on location effects meta-analysis. And this is because we have 2 specific community standards that allow us to sort of retrospectively look at consensus across brain imaging studies. And one is that we typically all spatially normalize our brains to a standard template. So we're all talking about relatively the same brain space with, you know, a little asterisk there for the complications across different templates. And that typically papers are full of results being reported as 3D coordinates, those XYZ coordinates that represent the peak locations of the activation blobs. So in terms of meta-analysis methods, in 1997, Peter Fox and colleagues developed a quantitative method for assessing regional convergence. But that particular algorithm, again, 1997, didn't really take root. Also in 1997, there was a really cool paper that came out by some folks at WashU, led by Gordon Schulman. And they noticed that no matter what type of task was performed in the scanner, a consistent set of regions were observed when they look at decreases, and that is which regions exhibited higher activity at rest than during task. And so reading that paper can be kind of fun. And then 4 years later, of course, that landmark paper by Marcus Rakel and those same people at WashU gave us the default mode network. So this idea of looking back at our results and establishing consensus and looking for trends and looking for patterns has been around for quite a while. the work that was done then in the early 2000s, that started this trend of launching quantitative formal meta-analyses. And an important paper around that time was one by Peter Turkeltaub and colleagues at Georgetown University. And that was the one that established the activation likelihood estimation method, which I'll be telling you a little bit about later on. So let's talk about why we're interested in meta-analysis in the first place. And the reality is that there are limitations of neuroimaging studies. Historically, we've been dealing with small samples for quite a while, especially compared to other fields of cognitive and social science, especially compared to the clinical domain. The BOLD effect in particular is an indirect measure of neuronal activity, and its reliability is limited by biological, technical, and methodological confounds. We also have a lot of publications that deal with isolated findings. It's expensive to do neuroimaging, so additional experiments that look for replication and confirmation and extension, those are rare because a lot of folks just simply can't afford to do them. And then we also have a habit of generalizing our context-specific findings, and Tao can tell you quite a lot about that. In general, our literature is full of studies that are making inferences about brain function and pathomechanisms that are based on a specific observed difference between two conditions in the scanner. So for all of these reasons, there are limitations to neuroimaging studies, but we have a lot of studies. So my sort of career philosophy has been, let's see what we can learn, what we can mine, what we can harvest from that literature. So again, thanks to these two community standards, the spatial normalization and the fairly high level of reporting as 3D coordinates, we do have a lot that can be mined. And so one particular form of meta-analysis that is very common is a coordinate-based meta-analysis. And that asks the question, where do the foci converge? So if you dig around in the literature a little bit, you will see tables and tables and tables of coordinates. The idea here is that we can extract those coordinates and plot them on a standard template and look for where they converge spatially. Again, coordinate-based meta-analysis is one of the basic two types of meta-analysis. Activation likelihood estimation is a popular choice for coordinate-based meta-analysis. There are a couple of different options out there for you to choose from. I'm going to give you an example with AL as the basis. So let's walk through what this is about. The general approach is that you convolve your foci or your XYZ coordinates with spatial kernel, here a 3D Gaussian distribution, to produce study-specific modeled activation maps. The point is that you can pull those XYZ coordinates from the literature, but then you got to kind of blur them and inflate them back up to reapproximate the original statistical map that they are extracted from. And then you want to combine those modeled activation maps into a sample-wise map to have some sort of estimate of their consensus. And then through a variety of different approaches, you can examine the null distribution of your test statistic to determine voxel-wise statistical significance, and bing, bang, boom, there you have a nice meta-analysis map. Okay, so as I mentioned, there are a number of different algorithms out there, and these were nicely summarized in a 2017 publication. Then, and there are pros and cons to various different methods, and I'm not really here to like advocate one versus the other. We use AL a lot simply because historically I have used AL a lot, but there are definitely pros and cons to all of these different methods. The second major category of neuroimaging meta-analysis is the image-based approach. And so here, rather than pooling the coordinates, IBMAs pooled the statistical maps, which have better spatial specificity than the peak coordinates. And so this type of meta-analysis asks, where do the images converge? Okay, so various different image-based meta-analysis methods are available. They were nicely summarized in a 2016 publication by Camille and Tom. I'll note here that image-based meta-analysis is preferred, given that additional information that is contained in the maps offer us compared to those single XYZ coordinates. However, image-based meta-analyses are fairly rare, and this is because a hardworking researcher can dig through hundreds of papers and harvest the coordinates from the tables, but they can't magically gain access to the original statistical maps when they are not part of the original research group that published that study. So this is why at this point in the presentation, I say, hey folks, please upload your maps to NeuroVault, even if you have no interest in meta-analysis, because image-based meta-analysis is the better route, but it doesn't really have a chance. It doesn't have a future unless all of us adopt this little bit of data sharing, this little bit of low-hanging fruit where you kind of say, okay, NeuroVault, here, take these maps from this analysis that I did. Until that makes it into our publication pipelines, image-based meta-analysis is not going to become a common method of examining consensus for us. Okay. All right, so that's my quick and dirty background into neuroimaging meta-analysis and the different approaches. And I like to get through that fairly quickly because it gives me more time to show you applications and examples so that I can better illustrate to you why meta-analysis might be useful and might be worth your additional consideration. So I like to think about these examples from the perspective of who you might be. So let's say you're starting out on a brand new research project, one where you're unfamiliar with the existing literature. You know you need to dig in and start reading, but sometimes reading habits can be all over the place, and you can go off on these little holes of exploration of the literature, which can be fun, but you know, all of a sudden then you find weeks have passed and you need to swing back around to what you're supposed to be focusing on. Maybe that's just me. Lots of times it can help to have a very clear and focused goal. In this case, meta-analysis can serve as an opportunity for you to read through the target literature in a systematic way while synthesizing those papers and working towards a quantitative outcome. That is a nice map that summarizes regional consensus for all of those papers that you just read. Second use case is very much related to the first, but it fills a unique gap on your to-do list. Maybe you're a grad student and you've been asked to mentor an undergrad, or you're just someone who's been asked to mentor a more junior colleague, and oh my goodness, they have no prior training in neuroimaging, so what are you going to be able to do with them? And sure, you could give them maybe some QC work to do, but what about helping guide them through a meta-analysis? Meta-analysis don't require the same sort of computational rigor that an analysis of original data does. So this is something that likely will be feasible given a novice and limited skill set. And like the first example, it helps them and you walk through a new literature. Okay, so next are some examples for more experienced researchers. Number 3 is fairly common. Say you have some hypotheses for a new data analysis and you want to focus your attention on regions of the brain that are associated with, for example, autographical versus maybe working memory. Head to the literature and grab either an existing meta-analysis or do one on your own and use those results to regionally seed your new data analysis in an unbiased and data-driven way. Number 4 is a little less common, but still kind of a cool option. Say you want to test out a new task version or data acquisition approach. You do your new thing and compare it to results to the standard published results in the form of a meta-analysis. And this can go a long way in discussions with reviewers towards validating your novel approach. The last 3 are a little bit more complex, so I'm going to walk through them a little bit more slowly. Number 5 here is functional decoding, so let's talk about what that is. Okay, let's start off with defining the term reverse inference, which you may have heard so far. I highly recommend reading 2 of Russ Poldrak's papers on this topic. One is from Ticks in 2006, and the other was a perspective in Neuron from 2011. Basically, forward inference is given the performance of task X, brain activity was observed in region Y. Reverse inference is given brain activity in region X, therefore mental function Y was engaged. Okay, so in like example words that make more sense, a fear task activates the amygdala. That's forward inference. Reverse inference is the amygdala was activated, thus fear was engaged. And reverse inference is the one that can be a little dangerous, right? That we don't necessarily know that fear was engaged. Maybe the person was happy. Maybe there was some other thing going on that activated the amygdala that didn't have anything to do with fear. So the issue is that reverse inference obviously sounds bad. When you phrase it like this, the amygdala was activated, thus fear was engaged. That's clearly not how we want to make inferences in our papers. But you know, it's something that can kind of sneak up on you in really subtle ways. The problem is that there are papers that have shaped my worldview of how I think about brain regions and their functions, and I think that that's true for everyone. We're not machines after all. We can't read all of the papers. So the papers that really resonate with us, stick with us, and inform how we think about certain regions and networks. And the way that we write our papers is that we run some analyses, either from an experiment we designed or that somebody else did, and then we interpret our results. And those interpretations, for better or for worse, generally fall along the line of, and in this result, we observe the anterior cingulate cortex, which is typically observed during conflict and response selection, or something like that. And so without knowing it, we're just trying to cite the literature that we know. All of a sudden, if you take a step back and look at your discussion section, it's full of reverse inferences. Okay, so what do we do about it? In those two Poldrett papers that I mentioned, Russ puts forth a way of managing reverse inferences from a data-driven viewpoint. Recent developments in statistical analysis and informatics have provided new and more powerful ways to infer mental states from neuroimaging data. And with this, we're talking about using a database approach. And there are two main databases here, the NeuroSynth database and the BrainMap database. Reverse inference can be a useful strategy if it is based on real data, such as meta-analytic maps, rather than on an informal reading of the literature. And so this is the key message here. And there are two papers that I would present as examples. One is that I was first author on way back in 2009. And the second is one more recently about Alejandro de la Vega from 2016, both of which use large-scale meta-analysis to do functional decoding, to do reverse inference, but to do in a way that is based on large-scale data mining and meta-analysis rather than this informal reading of the literature. So many large-scale meta-analytic databases they allow you to consolidate these results from many studies and annotate them to make them more searchable. The algorithms that these databases offer make performing the meta-analyses easier. Lots of people ask me, well, BrainMap versus NeuroSynth, what do I use? Well, they're different, they're complementary, they have different strengths and weaknesses. BrainMap was created back in 1994, NeuroSynth in 2011. BrainMap is based on manual annotations of the literature. So there are live human beings who are reading papers, extracting details about them according to the BrainMap coding scheme and entering into the database. NeuroSynth is based on automated annotations that pulls words from papers. The NeuroSynth is also much, much larger than BrainMap. So looking at what each of those databases offers you and what your goals are is generally the best way to go to decide which resource is right for you. Both offer tools for and methods for meta-analysis and decoding. So that's how meta-analysis can help with functional decoding and help you avoid the dangers of reverse inference. The next application that we're going to be talking about is meta-analysis as a data-driven approach to validate cognitive models. Okay, so as more and more resources are being deployed for the management, sharing, and meta-analysis of neuroimaging big data, and this includes knowledge representation systems or ontologies. But cognitive descriptors are relatively underdeveloped. We have lots of ways of describing image acquisition and data formats, and you'll be hearing so much about BIDs. We have lots of descriptors for lots of different aspects of our data, but the cognitive aspects of our data are relatively underdeveloped. And even getting a list of the paradigm or the mental function that was engaged by the contrast, those can be really, really challenging because there's not a whole lot of consensus in how we describe those things. So what I'll be describing to you is an idea that we came up with several years ago where we wanted to do some data mining and let the data facilitate some consensus. So the overall premise of this is that differences in activation patterns across studies should be captured and leveraged as they indicate potentially meaningful segregations in brain function, and that tasks activating similar brain networks should be grouped as functionally similar in a cognitive schema. And then tasks demonstrating differential activation patterns should be classified as functionally distinct. Okay, So what are we actually talking about here? Let's say we have a meta-analysis topic. We go and extract the coordinates from the literature. We generate modeled activation maps for each experiment or contrast or unit of these statistical maps. And then we generate a correlation matrix where we compare the spatial similarity of those maps to each other. And we perform hierarchical clustering of those modeled activation maps to identify clusters of experiments with similar patterns. So let me give you an example. We did this with face perception tasks, and we identified 4 different clusters. The first cluster, because all of this is mapped to a database with annotations, either manual or automated, whichever, but because we have metadata annotations of these experiments, we can look back to see which metadata fields drove these clusters. And we identify our blue cluster here as experiments belonging to visual spatial attention and visual motor coordination of faces. The activation pattern that was revealed in this cluster was resembling that of the salience network that includes bilateral insula and anterior cingulate. The second cluster, the purple one, the metadata trends demonstrated that these experiments were associated with perception and recognition of faces. The maps themselves demonstrated amygdala fusiform face area and occipital temporal areas. Our third one was associated with social processing and episodic recall of faces. And this map was strongly resembling the default mode network, including those lovely little medial prefrontal cortex and precuneus posterior cingulate areas. Finally, our fourth cluster was much smaller in terms of extent across the brain, focused on face naming and lexical retrieval, and included middle temporal gyrus, inferior temporal cortex, temporal pole, and parahippocampal gyrus. Okay, so we threw a whole bunch of maps into a bin. We ran hierarchical clustering. We got 4 clusters back, and each of those clusters had certain metadata trends to them. And the question that we wanted to ask next was, okay, well, so what? How does this help us learn anything about face perception? So we thought we would take a look at how well our current results align with an existing cognitive model. Gabini and Haxby published a model back in 2002. I think they've refined this model either both in the prequels and sequels of this 2002 paper. And what we saw when we dug into the model itself is that there was a core system associated with perception and recognition of faces that matched really well with our purple cluster. There was also multiple extended systems, one of which having to do with social processing and episodic recall of faces, just like our green cluster, with a little one embedded in there that matched up well with our face naming and lexical retrieval. And then there wasn't much mention of the salience network's involvement in face perception. And so we basically were able to confirm and extend a well-known cognitive model of face perception. And we demonstrated with these results that this large-scale data mining approach it's from an agnostic theoretical perspective is able to inform the evolution of cognitive models by meta-analytically probing the range of behavioral manipulations that are present across tasks. So that paper was published in 2015. We have used this model since then. This is Katie Bottenhorn, a grad student in my lab. She published a paper in 2018 that did the same sort of approach in naturalistic tasks. We have Cody Rydell, also in my lab in 2018, did the same approach with emotional tasks. Jessica Flannery, also in my lab, did this with reward tasks. And then we most recently had a collaboration with some colleagues of ours over in Germany that looked at emotion regulation. And so we're finding this method to be quite flexible and allow us to really kind of dig into a domain of interest. And for me personally, staring at these meta-analyses images for, goodness, 20 years or so, I guess 15 to 20 years, who knows? It really starts kind of reframing how you think about brain networks. And that, we did see in each of these different papers, consistent representation from the salience network, consistent representation from default mode, and also central executive frontal parietal processing. And this makes a lot of sense from that perspective of the triple network model. And this is the Menon paper from 2011 that posits the psychopathological processes. Those are very frequently characterized by disruptions in the salience network, default mode network, and the central executive network. And so looking at the interaction both within and between those networks, that's certainly becoming more and more common in the literature. And based on what we're seeing from a meta-analytic perspective, rightly so, these networks are consistently involved in certain large domains of interest, and therefore should be examined more closely from the perspective of psychopathology. Okay, so let's move on to from validating cognitive models to systematic variability, for example, in your participants and your tasks, and looking at how this can potentially resolve conflict in a given community. And this is something that we have done more recently. Speaking of those large-scale networks, if you spend any time reading through the neuroimaging literature, you'll see that a variety of names that all reference canonical frontal parietal patterns. You see central executive network, cognitive control network, dorsal attention network, executive control network, executive network, on and on and on. Lots of different labels have been generated to sort of characterize a very classic frontal parietal pattern. And we were curious to know if the current state of naming conventions for these commonly observed patterns, whether that's inconsistent or consistent through the literature, because if it's inconsistent, then that can be confusing, but it also can really limit our ability to coherently discuss and interpret our community's collective findings. So we thought we'd leverage image-based meta-analysis. Everything I've shown you at this point has been coordinate-based. We did a little image-based approach to explore whether these labels related to executive function, cognitive control, basically these frontal parietal patterns, whether or not they overlap or they're clearly separable in terms of different topographical brain networks. So to do this, we searched PubMed for papers reporting spatial patterns with the corresponding functional labels listed here. We focused on studies examining data from healthy participants, 18 to 70 years old, that used whole brain methods. We were able to grab a few maps reported from studies in NeuroVault, but for the most part, we had to email the corresponding authors to ask for their maps. This took many, many months, a lot of follow-up, a lot of emails. But in the end, we obtained 365 maps from 188 publications. So what did we find? We resliced and renormalized the maps. We performed a different series of image-based meta-analyses. And first we found that when you're looking at the label within one of these 9 categories, no individual label map exhibited correlation of greater than 0.6 with its respective constituent maps. So within a category, there was quite a lot of variability. Between the different labels, the average maps for EN, FPN, CEN, and WMN, those all exhibited a correlation of greater than 0.8 with the average of all maps. And then, so those were all very highly correlated. Then we had the VAN, ECN, TPN, CCN, and DAN. Those all had much less correlation. So multiple different labels are being used to describe topographically similar networks, but there is some evidence for distinct networks in other labels. So it's like, yes, in some cases, the labels are just a mess and they're all talking about the same thing. And in other cases, maybe there's some difference there. So we went ahead and put all of the maps into one big bin, ran clustering and analysis on it to see whether or not this really is one big network or if there is some grouping. And it turns out that, yeah, the labels are inconsistent, but the hierarchical clustering analysis of the images themselves revealed 4 distinct topographical clusters. So we have a preprint out that talks about this. It's fairly short. The big take-home message with us is that, look, labels are being used inconsistently. Image-based meta-analysis shows us that. We are advocating for a more consistent and coherent nomenclature to be used in papers that report activation or connectivity or whatever it is in these frontal parietal networks. Okay, so making a little bit of a transition here. I've given you 7 reasons why I think meta-analysis might be able to be helpful to you. There are various different methods that are available for you to do both coordinate-based meta-analysis and image-based meta-analysis. So maybe I've convinced you. What would be the next step? Okay, so first of all, there's a library of meta-analytic images called ANIMA. It's kind of like a meta-analysis database. And you can go navigate to that website there at the bottom to browse for ideas and examples of other folks who have been doing meta-analyses. Once you're ready to do your own meta-analysis, there are resources and tools available both at BrainMap and NeuroSynth. Again, BrainMap is for your detailed manual paradigm-based meta-analysis. NeuroSynth is for your automated and fast meta-analysis and decoding. At least historically, those have been your 2 options, okay? Currently, all of the methods you see there in the middle of the page, those have been spread out across different programming languages and user interfaces. So what I'll be talking about next is the new approach that we are promoting. And that is something called NIMARE, a neuroimaging meta-analysis research environment. The goal of NIMARE, with its super cute spider logo, is that we want to implement a common interface across different meta-analysis packages that perform different tasks. And this is also part of our open source community development of software and tools and resources. So by doing so, we are hoping to provide a standard syntax for a variety of different analyses that handle both coordinate-based and image-based databases and meta-analysis methods. And so NIMARE is joining the Python neuroimaging ecosystem along with a number of tools that you'll be learning about as part of NeuroHackademy. I mentioned Taylor specifically as he has been the driver of NIMARE and has absolutely devoted his time and energy to creating an amazing resource for everybody. So let's talk about the role that NIMARE plays in the broader Python ecosystem. So all of the meta-analysis methods that I mentioned before are spread out in a bunch of different programming languages, user interfaces. Some of them are never even translated from the papers to usable tools. And so that is what Taylor has been working on for the last few years. This slide here is intended to represent how NIMARE fits into a lot of the other Python tools that are being developed for neuroimagers. And so right now I know Tala is working very hard on NeuroSynth 2.0. And this is just intended to give context for how NIMARE is going to fit into all of that. So at this point, I want to make a little bit of a transition. I do want everybody to check out NIMARE. Feel free to contribute to it. We'll have folks available if you want to talk about that as we move forward. But in my last little bit of time here, I want to make a bit of a transition. I thought about pulling these slides out of my talk, but given some of the questions and discussions that I saw yesterday, I think that they might be a little bit helpful. Discussions about open science and reproducible strategies are becoming more and more mainstream, especially if you're on Twitter. But that does not mean that all labs have the same philosophies, and they certainly haven't all embraced open science in that way. So hashtag not all labs, right? So for the next few minutes, I'd like to walk you through some of the practices that my lab has adopted over the last couple of years. And some of these are best implemented as a lab-wide approach, but others you may be able to choose to do for yourself, regardless of your lab's general outlook on these issues. And you may even find yourself stealthily changing the culture of your lab by just starting out with a few of these ideas. And to be honest, that's exactly what happened in my lab, thanks to people like Taylor Salo and Katie Bottenhorn. Okay, so first is one of the big ones. As a lab, we decided to implement a standard workflow for moving our images from the MRI console to XNAP, which is our long-term image archive, to the high-performance cluster, the HPC that we use at FIU. Lots of acronyms there. From there, our DICOM images are automatically converted to BIDS images. You will hear more about BIDS tomorrow from from Kirsty. And then we use MRI QC for quality control and fMRI prep for pre-processing, both of which you'll hear more about tomorrow. In some ways, this is the easy part, the beginning of our pipelines where we're getting data from the console and we're getting it ready for pre-processing and doing the pre-processing. this part can be standardized. It's the after the pre-processing that things can get a little hairy. There's so many different ways to analyze data. So yeah, see that NARPS paper again that was mentioned yesterday. So let's talk about those analyses. And yes, they're going to vary from paper to paper and from lab to lab and person to person. But all of your neuroimaging analyses are going to require some sort of code. And most psychology research does, because GUIs are not reproducible, and there are these proprietary tools that act as barriers for people without the resources to buy them. So even if you write very simple code like bash scripting, you will want to manage that code. Why? Because as projects age, we lose our code, it becomes outdated, or we forget how it works, and method sections are unable to fully convey all of the steps and settings we use for a given analysis. Okay, so this is just basic, like you're gonna need to reproduce some things, propaganda, right? Several code management tools exist. You have heard all about Git and GitHub from Elizabeth this morning. We absolutely have adopted GitHub. It makes it easier to contribute to existing neuroimaging tools. It makes it easy to take our new work and integrate it with existing stuff. Frankly, it's also the best funded, so it's easier to use and it has all of the bells and whistles. We manage our project-related code in private repositories while we're working on them, and then we make those repositories public when we submit our manuscripts, so that the manuscript itself can include a link to the relevant code. So if you haven't already, please head to YouTube and watch Elizabeth's video from this morning about Git and GitHub. Okay, so you collected your data, you analyze your data. Those are all the things that you do that you control. What about the other stuff that other people are doing that you can take advantage of? And so here is my little quick cheat sheet list of data and software resources with a quick definition of what they do. You've already heard about OpenNeuro, which is an image data repository, but again, also have a strong look at NeuroVault. I'm sort of hyper pro NeuroVault these days because we've recently attempted that image-based meta-analysis and realized that, come on, we all need to be uploading our maps to NeuroVault. This is just a few list of my favorites and a good place to get started if you're trying to connect with the broader neuroimaging open science community. So for a bird's eye view, we endorse using open source bids-friendly tools for data analysis. Once we get our results, we make sure the code is available on GitHub, that our maps have been uploaded to NeuroVault, and then sometimes, not always, I'm not always as good at this, making sure that our figures have been uploaded to Figshare. And then all of these things get referenced in the paper. As you're writing the manuscript draft, before it's a preprint, before you submit it for peer review, make sure those links to your repositories, to NeuroVault, are all in the paper. That way people can access them when the paper is published. And then while not all labs endorse doing this, and so if you know, this may be something that your lab culture does not support, but if you are ready, you can share your image data with OpenNeuro and write a data paper, which is cool because then you can then be cited by others who use your data for their research. So this is sort of our bird's eye view of the different ways that we touch open science and reproducible neuroimaging. Okay, but let's talk about those manuscripts and papers. Russ talked a little bit about pre-registration yesterday, and I'd like to emphasize that yes, register reports are awesome. And yes, we need more of them for many different reasons. But if you aren't able to go the register report route, you should still consider developing an analysis plan prior to starting any analysis and registering that plan. These documents are basically your intro and method section where you describe the rationale for what you want to do and why, lay out your hypotheses, and then write out the methods you'll carry out to test those hypotheses. And in my lab, this is highly recommended. It's mandatory for anyone, but especially for trainees, as a method to communicate in advance with your mentor exactly what you want to do. So you can iron out any of those issues before you start the analysis and avoid that endless cycle of meeting with your advisor and showing them your results and going back for one more thing. And then, well, what about this? And what about that? And oh my God, we've been doing analyses for how many years? This lets you come together and decide what's going to be done before you start touching the data. The pre-registered analysis plan can be privately uploaded to OSF. Nobody else has to know about it or have access to it. This is the Open Science Framework, where it will have a link associated with it. When you finalize your analysis plan, perform the analyses, get your results, then you pull out that plan and wow, you've already written your intro and methods. That's half your paper done. So now you write the results and the discussion. And the big point here is go ahead and tell everybody in your methods section that you pre-registered your analysis plan, put that link in. So this isn't the same as a register report in which you get actual peer review and feedback before you run your analyses, but it's better than just jumping in without really thinking about what you're doing and how you're going to do it, which is how many of us operated for years and years and years. So for our lab, we've been working on and plan to submit a register report for replication and extension soon. We've also fully endorsed the preprint approach. We've released several preprints within the last couple of years. My goal now is to make sure we release preprints for all of our manuscripts and at least pre-register our analysis plans for all projects with a confirmatory analyses. So for some of my more novice students in my lab, this analysis plan, it's tough the first time you sit down to write one of these, and there are some struggles. But it's better to work through that before you start messing with the data so that you are on equal footing with your mentor and everybody understands what everybody's doing. And so there are a number of different preprint servers once you get to that stage, like bioRxiv. Those integrate with Google Scholar and ORCID so that your preprints are logged automatically. Okay, so that's sort of it for my topic about meta-analysis and reproducibility. One last thing before we go. I want to say a little bit about our community interactions. I believe that it is critically important that we normalize talking about our community while we're talking about our science, not just relegate these comments and discussions to satellite events. Academia is very hierarchical. Power differentials, they create space for the abuse of power. And this is true irrespective of many different aspects of our identity. And so a lot of times you'll go to events or you'll have discussions or you'll meet with someone one-on-one and advice will be presented, and it can be really well-intentioned and come from a beautiful place, but it can often be really, truly terrible. And so in the cases of abusive powers, if you get mentoring on this, you're often told, tell someone, report their abuse. And this can backfire. Sometimes nothing is done. Sometimes there's retaliation. Sometimes this results in a lot of negative impact on one's career. Sometimes you're told, it'll get better, I survived, and so can you. And so what, I'm supposed to just deal with it? Okay. Find a community of support. Find a support group out there to help you get through this, which, yeah, you kind of absolutely should, but that's not great advice from the perspective. It's not the victim's problem to fix or cope or manage this, right? So the point that I want to make, and the reason why it's important, I think, that I make it here and not in a session on diversity and inclusivity, is because a toxic climate hurts everyone, and it is our job to collectively do something about it, right? So what can you do? You listen, you don't interrupt, and you prevent others from interrupting. You seek out other perspectives. And this one I have found to be absolutely key. I am a white Gen Xer woman mom. That's my identity. And so it's important for me to find other perspectives that are not linked to my own identity, that I can learn from their perspectives and get a better sense of what the world is like more collectively. So follow your LGBTQ+ scientists, follow Black, Latinx, Muslim scientists, scholars, and activists. Twitter's a nice place because people will tell you things and they will do the emotional labor of giving you their perspectives. All you have to do really is follow them and listen to it. And then more actively, change your surroundings, situate yourself as another. If you look around and the people that you're interacting with all look like you, all talk like you, and all have your background and perspectives, maybe you should seek out some diversity in your interactions with your collaborators. Because by seeking out like-minded people, we're creating our own scientific echo chambers. And rich and innovative ideas are cultivated in a really diverse and inclusive environment. And there's a PNS paper out recently that talks about just that. So this is something that I think that all of us should be striving to improve our community. And this is something that regardless of our background and our perspectives, it's something that we can do to help each other. And between the recent mainstreaming of the Black Lives Matter movement and the experience that I had recently on the Open Science Room at OHBM, I am more hopeful than I have been in years past. And I'm very excited about the youth and energy and inclusivity of the next generation of scientists. Okay, so with that closing bit, I will say that one thing that has meant a lot to me is my service at Florida International University. We are a minority serving and Hispanic serving institution, and working and being part of an institution that is so diverse and inclusive has really meant a lot to me and provided me career fulfillment in a way that I didn't even know that I was looking for. So as you're all moving out onto the job market, please keep an eye on your HCBUs, on your MSIs, on your HSIs, on your PBIs. There are all these different acronyms that are meant to designate institutions that serve students who are traditionally underrepresented, particularly in STEM fields. Okay, one last slide, and then I will stop for questions. And that is to announce, and Ariel did give me permission to do this, I have recently been funded along with the ReproNim team that includes David Kennedy, Satra Ghosh, and JB Poline to develop a course on reproducible analyses of the ABCD data set. ABCD study is the largest longitudinal study of child development in the US, but the data can be kind of complex. So we are going to walk everybody through all of the different resources and data types that are available by ABCD and do that hand in hand with teaching reproducible neuroimaging approaches. There's going to be a 13-week online course followed by a 5-day project week where we will work on collaborative data projects of the ABCD data set. Please have a look at the website. I'll post this, I think, to the random channel later on today. But we are definitely looking for not only student applications, for students, postdocs, early career faculty who want to take the class, but we're also looking for teaching assistant applications. You'll get a $5,000 stipend for working with us over the course of the course and helping out with our students. And so all of that is present on the website. And at this point, thank you for your time. And let me flip over to questions if I can figure out how to do that. Okay. I'm a psychology grad student new to processing neuroimaging data, a lot of great information. That's in the wrong channel.
I think probably you'd see like there's a Q&A thing at the bottom in Slack. Although people could also ask questions. And I guess sorry, I'm monitoring Slack.
No questions so far that I can see. So anybody got anything? How are we doing? How's NeuroHackademy going this year? Oh, we have a question. How is this different? Several studies using it. Okay. So, all right, a standard meta-analysis that I glossed over super quick for sure is let's start with something super easy. I'm interested in the Stroop task. I want to know what the Stroop activates. So I take all of the coordinates that have ever been published about Stroop task versus control, and I load it into an algorithm like ALE, activation likelihood estimation, and I get a single map out that says this is consensus for Stroop activation across studies. The MAG approach that I showed is simply saying, okay, look, there might be some variability across all of those different sets of coordinates that you included in the meta-analysis. So let's look for patterns in terms of groups of maps that are more similar to each other and different across those clusters. So it's just a more advanced approach to tell you a little bit more information about the underlying patterns in your data.
Other questions?
So can you use the same meta-analysis techniques for connectivity and activation data in fMRI? So the connectivity question, I think there are some folks recently working on this. I haven't worked on it primarily because the way you define regions of interest can vary so much. So if let's say you want to do a meta-analysis of DLPFC connectivity, if you get 5 maps in there and they all define DLPFC differently, and maybe they all define it like in ways that are not just like different, but like not even the same region, then, you know, what are we saying about the consensus across maps? So connectivity is a little bit more tricky simply because it's a little bit harder to handle that ROI definition variability. But I want to say I saw something recently that somebody's starting to do work on this. I don't know any more than that. Sorry, Kendra. Okay, so the difference in the, okay, so how is that difference defined? And that links us back to the MAG approach. Okay, so if you have like a bucket of maps and you throw them into a hierarchical clustering algorithm, you will get back clusters of different maps. And those will be different in the sense of they have different spatial patterns. So the difference that we were talking about in that particular slide is the difference in spatial patterns across clusters of maps that are all flagged as examining, let's say, emotion regulation or naturalistic processing. And then we look at those different clusters, we map back to the metadata, and we say something meaningful about the trends in metadata that map to those differences in clusters. All right. Oh, yeah, this is a good one. How do you deal with ROIs and meta-analysis? So we don't. That is usually one of the exclusion criteria in any meta-analysis, is that you can do let's say you want to do a meta-analysis on math processing. You'll go do a lit search. You'll start doing one of those lovely little Prisma charts that walk you through the different inclusion-exclusion criteria for your meta-analysis. Let's say you exclude studies that don't include healthy participants. You'll look at a particular age range so that you don't want to include results from children or from the very elderly. And then you'll also exclude studies that report results that are ROI results, where only one or two or a small number of regions were analyzed from an a priori perspective. What we're looking for in meta-analyses is the inclusion of coordinates that are the result of whole brain analyses, so that we can, you know, as you say here, we don't want to unintentionally inflate consensus or convergence in a region like the amygdala simply because, you know, half the studies only looked in the amygdala. So those get excluded. Oh, and then somebody put the nice little comment. Nick Crossley has done the functional connectivity mapping. Yes, thank you for that. My brain is dead, y'all. Internet went out of my house. I had to go to campus. Had to present an ABCD talk right before this, so whew.
We appreciate it.
The age of Corona is rough.
Although, but you know, like, is it rougher though than like getting on a plane coming out to Seattle? And,
No, but then you're dedicated to the event, right?
Yeah, and you're in Seattle in the summer, which is better than probably both Texas and Florida.
Yeah, for sure. Like I always thought I hated travel, and so by not doing anything the last few months, I'm like, oh, I feel like I don't even know what year it is, what month it is. Like you use travel and you use events to sort of mark time and to annotate your life. And so anyways, thank you all for listening. I appreciate it.
We'll all get to it. Thanks a lot, Angie.
Alrighty, have a good time.
See ya.